Hi, my name is Kasper Roet, and I'm the CEO and co-founder of QurAlis, a company that I started together with two Harvard professors to develop therapies to stop ALS. My background is actually in spinal cord repair, paralysis. I worked in the Netherlands Institute for Neuroscience, where I did my PhD to develop a gene therapy to effectively, the intent was to cure people that were paralyzed. My lab was working on ALS as well at that time, but I thought ALS is not a disease that we understand well enough to develop therapeutics. There was, at that moment in 2006, when I started my work there, only SOD1 identified, which explains ALS in 2% of the patient population, but it didn't give us enough of an understanding of the full disease. And so I didn't know how to actually make effective therapies for ALS. I then moved to J&J, where I worked in the Janssen Vaccine Institute in Leiden to develop a new vaccine for polio. Polio is a virus that also causes paralysis. While I was working at J&J, I actually also worked at the Dutch Brain Bank and helped with organizing autopsies for patients with neurodegenerative diseases, including ALS. And that really made it even more clear to me how impactful ALS is and other neurodegenerative diseases are for patients and for their families. And I really wanted to do something about that. It was kind of the time post the Human Genome Project where a lot of transcription data sets are published, which are data sets that look at different messenger RNAs, mRNAs that are regulated in diseases. And so for many different diseases, including ALS, a lot of these data sets were put online. And so what you could actually do was look at postmortem tissue and look which genes are regulated in a specific disease, for instance, in the spinal cord of an ALS patient. And at that time, while I was working there, C9orf72 was discovered, and then rapidly other genes were discovered as well. And now there is between 40 and 80 different genes that are identified to cause ALS. That got me really excited about understanding of this particular disease, and I thought we can make therapies that are impactful. And so back to these mRNA data sets, what I did then, I was collecting all of these big data sets, and I was trying to find really important genes using the genetics insights that we have, using these big mRNA data sets from post-mortem tissue from patients to see if we could understand these disease and if we could find therapeutic targets so that we could develop a therapy. I realized we need a model system to also test these therapies. And I looked at data sets that were available from mice and from rats, ALS models, and I thought if I can find disease patterns that are identical in humans, in patients, ALS patients, and also in ALS models, then I may be able to identify not just what is going wrong in the disease, but also be able to test them. I was very frustrated when I found out that in the end, there was no overlap between the animal data from the rodents and the human data. And I was a little desperate because I thought I had a good way into developing therapeutics, but I had no way to test them. I then discovered work that was happening in Harvard, where a really amazing professor, from my point of view, Kevin Egan, collaborated with an ALS clinician in Mass General Hospital. And together they basically created the first motor neurons from patients. They took skin cells from an ALS patient and made stem cells, induced pluripotent stem cells from those skin cells. And then Kevin and his lab made motor neurons from those stem cells. And so for the first time, we could actually study what is going wrong in the motor neuron of an ALS patient. And if we find out what is going wrong, then we could also test potential therapeutics to correct that. When I saw that, I was like, oh my God, this is the missing link that we need from genetics to be able to bring therapies to patients. I want to learn how that technology works. And so I moved to Boston and started to work together with Kevin Eggen and his collaborator at the Harvard Medical School, Clifford Wolf, to really learn this stem cell technology in order to able to use this technology to develop therapeutics. After 3 years, we decided to start QurAlis, really based on this technology and based on really groundbreaking insights that were made using these patient-derived motor neurons and genetics into how the disease works, into how to develop therapeutics. So QurAlis was created with our first program, which is a Kv7.2 program, or as at QurAlis we call it QRL 101. It's a very promising strategy for ALS patients. What Kevin discovered is that loss of STAthmin2, that microtubule stabilizer protein, is also driven by missplicing event. And the missplicing is linked to TDP43. TDP43 pathology is present in about 97% of all patients. And so it's a very relevant biology for almost everyone that is developing ALS, everyone except for patients with SOD1 or FUS mutations. And this missplicing event leads to a loss of STAthmin. Loss of STAthmin leads to neurodegeneration. And if you bring back STAthmin levels, you can protect against that. And we have shown that in our preclinical models, in our stem cell models, with motor neurons from patients. And we also see that, if you knock down or knock out STAthmin in different mouse models, you see this loss of innervation. So we have developed a compound to correct for the missplicing. It's an oligonucleotide. We call it a splice switcher ASO. It basically restores back normal mRNA levels of STAthmin2 and thereby also normal protein levels of STAthmin2. And we are currently testing this in a dose range finding a trial in 6 different countries. And so we are very grateful for all the patients that are working with us there. We think that this is going to be a really impactful therapeutic. And the patients that we are working with are from Canada, the United Kingdom, Netherlands, Belgium, and Germany, and Ireland. And we're very grateful for all our partners in this work. And then we have a third program on a synaptic vesicle release protein, which is a protein that is very important for how cells communicate together, which is also not functioning, or the function is lost in ALS. And we've developed, again, a splice switching antisense oligonucleotide to bring UNC13 levels back to normal. And we have actually partnered with a large pharmaceutical company called Eli Lilly on bringing that program to patients as well. And that is an ongoing effort between now QurAlis and Lilly. So with that, QurAlis has 3 different programs on genetic targets for sporadic disease, Kv7.2, which is called QRL 101, STAthmin2, the microtubule stabilizer protein, which is called QRL 201, and then UNC13A, the synaptic vesicle release protein, which is called QRL 204. And so we are not a company that is implanting stem cells. We use stem cells as a neuronal model, so to understand disease and to test potential therapeutics, and that is really what we believe stem cells are really powerful for. I'm just very grateful for all of the patients that we are working with, and I really want to make sure that we work together in the development of these therapies. I believe that based on all of the genetic insights that we have, based on the technologies that we have developed, the stem cell technology, but also the technology to correct RNA levels and restore RNA levels, as well as small molecules to activate channels and restore function, that we are in a good position to develop therapies that are meaningful and will have a really an impact on patients' lives. So I want to thank you for your time. And the company is based in Boston, Massachusetts. So if you're ever looking for me, please do come to Boston, and I'd be very happy to meet you. Thank you. Bye-bye.